Bismillahirrahmanirrahim. We start another paper, paper 5. This is the March 23, paper 5-2, and this is the second video. I have completed some general tips in the first video, and the question 1 also partially has been done in the first video. Now, in the last video, I left off here, and I will start with the question 1D, which was, I finished that part and we're starting it in this video. Part D says, in a further study, the scientists investigated whether gamma and a hot water treatment reduce the acrylamide concentration of potato chips. Remembering now acrylamide is toxic and all that, which is from the past, from the question which we read earlier. 4 bags of one variety of potato were each exposed to a different dose of gamma radiation. So we had 4 bags and each was exposed to a different radiation. So 0 joules, 50, 100 and 150. So in your mind, you have to now think of this, that we have 4 bags. And you can do this rough work on the paper. You're allowed to do it. One got no radiation, one got 50 radiation, one got 100 radiation, and one got 150 radiation. So it's increased very properly. It's increased by 50 each time. It's not sort of just like irrationally done, like 50 to 80 and then 170 and then 250 or something like that. After exposure to gamma radiation, the potatoes from each bag, so in a bag there would be many potatoes, remember that. After exposure to gamma radiation, the potatoes from each bag were cut into 5 centimeter blocks. So we have 1 into 5 centimeter blocks. Potato blocks from each bag were given 2 different treatments. So, okay, 4 bags, given the gamma, 0, 50, 100, and 50, and then we took out the potatoes. The potatoes were taken out, and then they were cut into 1 into 5, and potato blocks from each bag were given 2 different treatments. So mentally, you should be able to visualize this. What are we saying? 1 centimeter into 1. What does that mean? And if you look at this one here, what does this mean when you say 1 into 5 into 1? And so mentally, you should be able to visualize this. I'd like you to pause the video here, have a look at it, and see if you can understand it and comprehend it. Now, we had cut these into potato blocks. Now, in the treatment 1, which you have to understand, is 100 grams of potato blocks. So we didn't take out all the bags. We didn't take out everything. We only took out 100 grams of potato blocks were cooked in 1 dm cube of sunflower oil at 170 degrees Celsius for 5 minutes. So they've given you exact how much oil did we take, at what temperature was the oil, and for how long did we cook them. So 100 grams of the potato blocks were cooked in 1 dm cube of sunflower oil at 170 degrees for 5 minutes. But the treatment 2 was slightly different. Treatment 2 was again same 100 grams. The 100 grams is the same. 100 grams here, 100 grams here. Of potato blocks were heated in 500 cm cube of water. Here it was 1 dm cube of sunflower oil. I'm just telling you that you read the question so carefully. This may not be relevant in this question, but it will be relevant in another paper. So were heated at 500 cm cube of water at 85 degrees Celsius for 5 minutes. This was 170 for 5 minutes. Removed from the water and then cooked in 1 dm cube of sunflower oil at 170 for 5 minutes. So that is the same. So in this it was just the 1 dm cube of sunflower at 170. Here it was first in 500 cm cube of water at 85 degrees Celsius. First it was 500 cm cube of water for 5 minutes, and then the same treatment as treatment 1, and exactly the same treatment, 1 dm cube of sunflower oil at 170 for 5 minutes. Now you have to read it just like I'm reading this question to you and I'm explaining things to you. You need to read it as if you're explaining it to me in the exam. And you can imagine me sitting there and you're explaining it to me. So if you visualize that, I think you'll be able to do a better paper 5 with the fear that I'm sitting in front of you. The scientists measured the acrylamide concentration of the potato chips after these treatments. So when we've done the treatment 1 and the treatment 2, then we measured the acrylamide concentration. This procedure was repeated further 5 times to allow statistical tests to be carried out on the results. The results of the investigation are summarized in table 1.2. So first read this very carefully, then look at the table. Gamma radiation 0 treatment 1, 4551. What was this? Mean acrylamide concentration of potato blacks in microgram per kg. So in the zero gamma radiation, it was 4551, and in treatment 2, it was 1768. So there is definitely, with no gamma radiation, there is the first treatment was not so effective because the acrylamide is more. Here, the acrylamide is less in the treatment 2. Then when we gave them 50 gamma radiation, it was 3629 in the treatment 1, but it was definitely less in the treatment 2. So that means putting first boiling them in water and then boiling them in the oil was more effective. Then 100, it is again 3310 and 1339. In 150, as the radiation has increased, you can see in treatment 1 also the acrylamide has decreased. In treatment 2, the acrylamide has decreased, but when you can look at it, it was basically, you look at it how much it has decreased. Now it says using the table in table 1.2, calculate the percentage decrease in the mean acrylamide concentration of potato blocks given treatment 2 compared to potato blocks given treatment 1 for gamma radiation at zero. So what are we going to be talking about? Now, this is what you have to comprehend and understand. Now, whenever you're doing such a question, you've got to first look at it. What are the things that we're looking at? It is zero. So basically, it's at 0 kg. Now, this is 4551 and 1768. Now, it says calculate the percentage decrease in the mean acrylamide given treatment 2. How much did it decrease? So, from 4551, it is 1768. So, 1768 minus 4551, or you could have also done it the other way around. So, and then you divided it by 4551 and into 100. So this comes out to be 61 minus negative 61%. So basically, you have to look at it as percentage decrease. From 4551, it went down to 1768. So that we divided by the how much was it in the initially compared to potato blocks given treatment 1. Treatment 1 was 4551, so you compare it to 4551 into 100 and you get 2 marks for this. So you have to round it off. So it's better if you say just 61% instead of saying 61.1 or something like that. Now the results in table 1.2, the ones the results in table 1.2 are these. This is table 1.2. Now all this is, as they say now, they're going to tell you in the next part of the question, results in table 1.2 are shown in a graph on figure 1.5. Error bars have been added to show 95% confidence intervals. Now here is the graph. This is the graph, and you can see the the confidence interval, the ones which are shown here. Now, these are the ones which you are showing here. These are the error bars. This is the error bar here. This is the error bar here. And these are the error bars here. And this is an error bar here. Now you see they do not overlap. They're very different here. This is the error bar here on this point, and this is the error bar here on this point. Right? These are the error bars. If you don't know it, please Google error bars and have a look at it. What does all that mean? Now the next part does what? It's a question on those error bars and on that graph. So the scientists concluded that a combination of gamma radiation and hot water treatment is the most effective way to reduce the acrylamide concentration of potato chips. Explain how the information shown in table 1.2 and figure 1.5 supports or does not support this conclusion. Now, it's a very favorite question of paper 5. They give you data and then they say, okay, does it support or does it not support? And you can see that all across the board, there are certain answers which you can write in everything. So it is which ones? Table 1.2 and figure 1.5. So this is figure 1.5 here. This is figure 1.5. And table 1.2 is on this page. This is table 1.2. So you look at them both again. You again look at it very carefully, and does it support? What is the question that they are asking you? They're saying, okay, the scientists concluded that a combination of gamma radiation and hot water, which was the treatment 2, and the hot water treatment is the most effective way to reduce the acrylamide. So you don't give me something which says yes it is and something which says no, no, but then this is wrong about it. I want you to listen very carefully and understand how you have to analyze the data. Now, the first point in which you support this is you've got to talk of what? This was zero radiation. This was treatment one. So now we are looking at treatment one with no radiation, treatment two with no radiation. Now, when we give it some radiation, now look at 50. In 50, when we gave it some radiation, now it has decreased to this. And from this, it has decreased to this. But you have to understand that the gamma radiation and hot water treatment, treatment 2, decreases the acrylamide concentration of potato blocks more than gamma radiation only, which was treatment 1. Either gamma radiation only or treatment 1. So treatment 1, it decreased to 4551, but in treatment 2, it decreased to 1768. This is the mean acrylamide concentration of the potato blocks. With zero radiation, what was it? After 50 radiation, what was it? So look at the table very carefully. Then if you look at this, error bars of treatment 1 and treatment 2 do not overlap at all doses of radiation. Error bars of this were here. While the error bars for treatment 1 and treatment 2 do not overlap. So this is treatment 2. Let's look at the error bars here. Now these error bars and these error bars, they do not overlap. Now the third point which you could have said is hot water treatment and 150, the highest gamma radiation dose, gives lowest acrylamide concentration. So we go back to this table. Hot water treatment and 150. So you see this is one. So you are always going to look at the lowest one. This was the lowest one. So at 150 gamma radiation dose and treatment 2 gave you. So you pick out the lowest figure and then you develop it on that, because that is going to support the hypothesis. So 1010 was the lowest, 1010 was the lowest. But when was the lowest? The lowest was when it was 150 gamma radiation and it was treatment 2, hot water treatment, and 150 gave you the lowest. So when you are looking at this table, you're going to look at the lowest figure and the highest figure and then try to conclude things from that. And the scientists concluded that a combination of gamma and hot water is most effective. Gamma and hot water is most effective, and it reduces the acrylamide concentration. Now it says, give me something which says it does not support. Now when you look at this data here, how is this data here telling you it does not support? Very simple. Here we had no radiation, and yet treatment 2 has decreased the acrylamide concentration. No radiation, but even though there's been no radiation, but treatment 2 decreases the acrylamide concentration. So that is the not support. That is the not support. I would advise you to have first done this paper on your own and then look at what I have to say about it. So when it says does not support, then you have to figure it out from the data. Where is something which is coming at okay, but we did not give any radiation, yet there has been a decrease. Yet there has been a decrease. So this was a point which you could have written is does not support. Now you've got to realize is that you have to give me 2 marks for support and 2 marks for does not support. Though this is only a 3-mark question, but then you might word something differently. So give me a 2 for that, 2 for that, or give me more for support and more for, then of course one of them would be correct. Then another thing which you have to understand is what else does not support. The fact that hot water treatment alone might be safer than gamma radiation. And then, of course, another thing which you can write nearly in everything, only one variety of potato has been investigated. Sometimes they use very few numbers, and the very sample number is very small. So in the does not support, you can say that. Here we only used one type of potato. We didn't say, okay, we took 10 different types of potatoes. So in the does not support, and then in the does not support, you can also say that no statistical test has been carried out, which does not show here. So there are some things which you can write in does not support. Number one, sample size is small, or you can say here only one type of potato has been investigated. Second thing, you can say that no statistical test has been taken out to prove or disprove a theory. That's why we use statistical tests. Then you look at this error bar. This error bar was for zero gamma radiation. So you see here, what do you see here? That error bars for treatment 2. These are the error bars for treatment 2. Error bars for treatment 2 overlap all gamma radiation doses, suggesting that decrease in acrylamide due to hot water treatment and gamma radiation is not significant compared to the hot water treatment alone, this one. In this we had zero gamma radiation. So that is the point. If somebody can understand this, it's a little technically a little tough, but then you can does not support, you could have given me the other points. If this is not very clear to some of the students, I know it's not clear to many of the students. This was a 20-mark question, and this was a tough question, but as long as you could do all those which you shouldn't be losing marks on, those are the points that are very clear. Some, of course, you have to lose some marks. It's not a very easy paper. You can't get full marks in this. Now let's go on to question. Now let's look at question number 2. Rice or Rhiza sativa is an important food crop. Rice plants are wind pollinated. Pollen containing the male gamete is transferred by the wind to female reproductive organs of rice plants. Fertilization and grain formation then occur. Weedy rice. Weedy rice is a wild form of rice that grows in fields of cultivated rice. We've got one type of rice, which is the one that we have cultivated, and in between we have some other chungered ones which are called the weedy rice. You don't want actually. Weedy rice competes with cultivated rice for resources like the new nitrates in the soil. Weedy rice plants are taller. So the ones which are the bad ones, they are taller. Weedy rice plants are taller than the cultivated rice plants and produce a low yield of rice grain. So these ones which we have planted, now they will give us less grain. So I as a farmer am not very happy because I don't want this weedy rice, but the other ones, they're reducing my main cash crop, which is the rice, so reducing that. Now, how have we resolved the issue? It said weedy rice plants are taller than cultivated rice plants and produce a low yield of rice grain. Now, several genetically modified GM varieties of cultivated rice have been developed, right? Several GM varieties of cultivated rice have been developed. One concern about the use of GM rice is gene flow from GM rice plants to weedy rice, and gene flow occurs when the wind carries pollen from the GM rice to the weedy rice. So we've got the one which we want and the one which we do not want, which are taller. So the pollen from this can go and land onto this. It says gene flow occurs when the wind carries pollen from the GM rice plant. So we've grown the GM rice plant. And from the GM rice plant, it goes to the weedy rice. This was the other one. The yellow one was the one we grew before. So what we have to understand is that we are now worried about this. Some scientists investigated gene flow from herbicide-resistant GM From the herbicide-resistant GM rice plants to weedy rice plants, the GM rice plants had a gene for herbicide resistance. Weedy rice plants do not have the gene for herbicide resistance. The scientists wanted to test the hypothesis that gene flow from GM rice to weedy rice decreases as the distance between the GM rice crop and the weedy rice increases. The scientists planted GM rice and weedy rice in a field as shown in figure 2.1. Figure 2.1 also shows that the wind normally blows in a, northwest direction. So we've got this diagram which we need to always look at it very carefully. Now this is the GM rice plant which is here, and then the rest of it is all the VD rice. The VD rice is all here, 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 and here. And this is a distance of 1 meter, 2 meter, 5 meter, and 10 meter. So the GM rice is in the center. This is the GM rice. And we're expecting the wind to blow in this direction. So we are expecting the wind to blow in this direction. And we planted them at a distance of 1 meter, 2 meter, 5 meter, and 10 meter. So this is the 1 meter here. This is the 2 meter here. And this is the 5 meter here. And this is the 10 meter here. Take your time, understand the question. After the plants had been pollinated and the rice grains had developed, the grains were collected from the VD rice plants only. Grains were collected from all VD rice plants growing 1 meter from the GM rice plants in the direction north, northeast, east, southeast, south, southwest, west, and northwest. Grains were also collected in the same direction from the GM rice plants at distances of 2 meter, 5 meter, and 10 meter. So 1 meter, there was more sides, and in the 2 and 5 and 10 were also collected. Then approximately 1,000 grains from each collection point were planted and germinated in controlled conditions in a glass house. The plants were grown for 3 weeks and then tested to determine whether they had the gene for herbicide resistance. Figure 2.2 shows the young rice plants 3 weeks after germination. Now identify two independent variables in this investigation. What is the distance? What is the thing that you changed? You changed the distance from the GM rice plants and the direction from the GM rice plants. These are the two independent variables that you decided to investigate. Number one was distance from the GM rice plants and the direction from the GM rice plants. Because you said, okay, let me see whether the pollen is found at these distances and in these directions. Northeast, southeast, west, northwest, and all that. Then part 2 says some of the young VD rice plants had the gene for herbicide resistance. The gene had been carried to the parents of these plants in the pollen from the GM rice plants. Outline a method the scientists could use in the glass house to determine how many of the young VD rice plants had the gene for herbicide resistance. Your method should not require the extraction of nucleic acid. Well, very easy. You apply herbicide to the young plants. Count the number of young plants that survive. When you apply the herbicide, if they are resistant, they will not die. If they are sensitive, they will die. It's very easy. How many people can you find out have respond to Panadol? Okay, so you take 10 people who have a headache, give all of them Panadol. 5 of them say we are better, 5 of them say we are not better. Well, the Panadol works on 5, Panadol doesn't work on the other 5. Panadol doesn't work on me either when I have my migraine. So apply the herbicide to young plants and count the number of young plants that survive. Then coming to part 3 of the question, the scientists calculate the percentage of young VD rice plant that had the gene for herbicide resistance. This percentage was used as a measure of gene flow. The percentage gene flow recorded at each NW northwest collection point is shown in figure 2.3. Figure 2.3 also shows the percentage gene flow recorded at each distance from the GM rice plant at all the other collection points combined. So here we have these graphs. This is the percentage gene flow. This is the distance from the GM rice plant, 2, 4, 6, 8, 10 meters. And we have the northwest collection points, which is this graph. And the all other collection points is this graph. So this is the one, which is this one. And this one is the NW collection points will be this one. So we can see how. Now you can see as the distance has increased, the percentage gene flow has gone down. At 1 meter, it was only 0.15%, and then it's gone down. At 10 meters, it is 0%. But in the all other collection points, it was only 0.03 or something like that, and then it has gone down to zero at the distance of 5 meters. So always read this graph to yourself and explain this to yourself how we're going to understand this question. It's a very important way that you understand. State two conclusions that can be made from the data shown in figure 2.3. Now, data in figure 2.3 is this. What is this showing you? The distance from the GM plant increases, percentage gene flow decreases. Simple. Distance increases, percentage gene flow decreases. Very simple, you have to write that. Percentage gene flow is higher. Where? In the direction of the normal wind, the northwest direction, in the NW direction. So here the percentage, so simple from the graph you have to just do this conclusion. It is higher here. It is not as high as this, so you compare this. So the percentage gene flow is higher in the direction of the normal wind, which is the northwest direction, as compared to the one in the other directions. As distance increases, percentage gene flow decreases. Percentage gene flow is higher in northwest direction than other directions. VD rice plants have a gene that results in increased height. GM rice plants do not have the gene. This gene allows gene flow from VD rice plants to GM rice plants to be investigated. In a second study, the scientists investigated gene flow from herbicide-resistant GM rice plants to VD rice plants and gene flow from VD to herbicide-resistant rice plants. The scientists planted GM rice and VD rice in areas next to each other in a field. After the plants had been pollinated and the rice grains had developed, 1,000 grains were collected from VD rice plants at each of the 8 sampling sites, and 1,000 grains were collected from the GM rice plants at each of the 8 sampling sites. Plants were grown from the grains that had been collected and tested to determine the percentage gene flow between the VD and the GM. Table 2.1 shows the results of the investigation, and you had to calculate the mean. That wasn't very difficult, so you just had to calculate this 0.036. That was the mean. So complete it by calculating the mean percentage gene flow from GM rice plants to VD rice plants. You added them all up and you divided them by 8, and that gave you 0.036. The scientists decided to carry out a t-test to compare the percentage gene flow from GM rice to VD rice with the percentage gene flow from the VD to GM. The scientists stated the null hypothesis: there is no difference between the percentage gene flow for GM rice to VD rice and the percentage gene flow from VD to GM. The scientists calculated the value of T as 9.043. Table 2.2 shows the probability table for the t-test. Explain with the reference table 2.2 what can be concluded from the analysis of the data collected by the scientists. Which degree of freedom are we going to look it up? So you see these are 8, N1 is 8 and N2 is 8. N1 is 8 and N2, so 8 plus 8 is 16 minus 2, so we're going to look up 14. At 14, the critical value is 2.145. So critical value is 2.145, 9.043, which we had found out which they had told us in the question, null hypothesis is rejected. Last part of the question, the results of this investigation are published in a scientific paper. A student who read the paper concluded that the results showed there were no reasons to be concerned about gene flow from GM to wild. Suggest one reason why the result of this investigation should not be interpreted in this way. So there are many points in this, but I'm giving you just few of the ones which I feel are very relevant. Investigation was only carried out once. Investigation was only done on one type of crop species, which are the wind pollinated crops. And then there is no data over several generations. Then the investigation is only in one geographical area. Investigation is only in experimental trials, and there is no assessment of significance of consequences. So a lot of points, but you have to give me one sensible point in order to get this 1 mark. And that completes this paper 5, and I hope everybody watches this video very slowly, very carefully after doing the paper, so that you can benefit in it in the way that in the exam you are able to handle any question. This question is not going to come again, but you should be able to handle any question which is coming in the exam very soon.